I learned about 6 months ago, I guess, that our biophysicists in physics were always conflicted because the what's the name? The structural and quantitative biology seminar was scheduled at exactly the same time as our physics colloquium, which was really, really unfortunate. So somebody had the clever idea that we should do a few joint of these every year. And so this is the first of those. So, and I'm really excited to see this going forward. So next week's physics colloquium is up here. It's Yuri Kolomensky, who's going to be talking about neutrinos. Today's speaker is Naji, and I'm actually going to hand the microphone over to Ran Shen, professor of physics, who is going to introduce today's colloquium. So.
Well, thank you all for coming to this colloquium instead of this breakthrough prize symposium that's going on, especially when our own faculty member, Kenbu Luke, got this year's prize. Congratulations to Professor Luke. It's my great pleasure to introduce today's speaker because she was one of my best students. And she got her degree at University of Science and Technology in China, working on a thesis using high field laser physics to calculate molecular light interactions. And then she came to Berkeley to do pioneering work on nonlinear chiral spectroscopy. And that's her PhD thesis. Afterwards, she went to Genelia Farm for microbiology to work with Eric Betzig, who got the Nobel Prize last year. But after a few years, she became the group leader at that institute. And then she went on doing neurobiology of the brain. I visited her lab once, seeing a mouse under the microscope with a head, with a hole on the head, with several wires going into the hole, and then shining laser onto the hole. Yak. It's certainly not the kind of research for me, but this is what she'll talk about today.
Thank you very much also for the invitation. I've been spending so many hours sitting in the audience listening to talks, so right now I feel rather stressed because I'm back in front of my advisor and going to talk about some yucky stuff. So also I want to apologize just in advance because I had a cold a month ago, but I'm still coughing. So don't be alarmed if I start to cough. It sounds a lot worse than it actually is. With that said, so you probably have been looking at this movie for a while. What you're looking at are the images of stars at the center of our galaxy. You can see that initially this image is kind of fuzzy, but if you use a wavefront shaping technique called adaptive optics, you can get a very sharp image of those stars. And you can then actually following their orbiting around the central supermassive black hole of our galaxy. If you think about it, it's actually rather miraculous that we can use a camera that is on Earth to look at something that is 25,000 light years away. But I would like to argue why you're thinking that something else equally miraculous is inside each and every one of us, and that is our brain. So our brain is not very big. It's about 3 pounds and 1,000 cubic centimeter. But it is where all our mental activity originates. And it's also the organ that allow us to interact and to understand the world around us. And if you look at into a small area, you will see that brains contain a kind of cell called neurons. So neurons has different type of morphology and shape, but in general, it can be separated into 3 parts. the part where the nuclei of the cell resides is called soma, or cell body. And of this cell body, there's many, many very fine processes emanating out. And the part of the process that receive information is called dendrite. And the part of the process that send information to other neurons are called axons. And when the axon of one neuron meets the dendrite of another neuron, they may form a structure called synapse. And that's where the neurons can communicate with each other. And usually the presynaptic structure, so on the axon of the first neuron, are called presynaptic boutons, while the postsynaptic structure on the dendrite of the second neuron are often called dendritic spines, because sometimes they often protrude out like a little dot on the dendrite. So neurons communicate via electrochemical signal. And this process can be monitored by measuring the membrane potential using an electrode. When the neuron is not sending any information out, we say that this neuron is at rest. At rest, the membrane potential is about negative 70 millivolt. And when the neuron starts to receive input from its dendrite, its membrane potential starts to increase until when it crosses a certain threshold, then the cell membrane potential will experience a very rapid change, which is called action potential. And this change can be propagated from the cell body of the neuron through the axon into the dendrite that the axon is making. And when the action potential invades this particular bouton, it will release the neurotransmitters that are contained in those vesicles. So in this way, information travels across the entire brain and gets transformed on the way and generates the wide variety of mental activities that is happening in every one of your brain at this moment. So I would like to argue that it is actually no less challenging to understand brain compared to when you want to understand stars. If you look at the numbers, they're about the same number. There's 100 billion, around 100 billion stars in our galaxy. And they are about the same order of magnitude neurons in our brain. But they make about, each neuron make about 10,000 connections. Even when you look at a much smaller brain, which is a mouse brain, the number of connections between neurons are still on the same order. It's 100 billion synapses. And it's not just about numbers, because consciousness, perception, attention, all this mental activity are actually emergent property of many neurons that form circuits through their synapses. So it is actually not enough to only study a few neurons. Just like if you only look at silicon atom, it's hard to understand how the band gap arises. So to have a nice technique that is capable of both studying the physiological processes on something as small as a synapse, which is about 1 micron, but it can also, the ideal technique should also be able to monitor the activity of many neurons in the circuit at the same time. And such technique actually exists, and they are optical microscopy. There's a particular form of optical microscopy that is very powerful for brain imaging, and it's called two-photon fluorescence microscopy. So in two-photon fluorescence, the molecule absorbs 2 photons simultaneously and get to the electronic excited state, and then can release 1 fluorescence photon and get back to the ground state. Because two-photon absorption actually requires second-order perturbation theory, the cross-section is rather small. As a result, it can usually only happen at laser focus, where the electric field intensity is sufficiently high. To generate an image, what you do is to move this focus across the sample. So here is the yucky experiment Ron was telling you about. You can open a hole in the mouse brain and replace it with a cranial window so that you have optical access. And then you focus your laser light into the brain and record the fluorescence intensity at each position. By raster scanning this focus, you can then generate an image of your biological sample, basically the fluorescence distribution in vivo. So the first demonstration of using two-photon fluorescence to image the brain happens about 25 years ago. And since then, its application in neurobiology has been tremendous. In one type of application, you can actually follow the generation of connections between neurons or the generation of synapses. For example, in this study, you could look at how the dendritic spine, which remember is a postsynaptic terminal, how they appear or in this case, I think actually disappear and appear when the animal is developing over multiple days. The same thing when the animal is learning a new task or when you're learning new knowledge, new synapses can also be formed, which can also be monitored with two-photon fluorescence microscopy. You could also look at the presynaptic part and look at synaptic botons. This example shows how the botton will appear and disappear during the aging process inside the animal. But what makes optical imaging particularly powerful is that it actually allows us to monitor activity of a neuron. You may remember I told you that the neuron uses electrochemical signal. How can we use light to probe its activity? So what I didn't tell you is that when the cell fires action potential, what happens is that the calcium ion concentration inside the cell will rapidly increase by about 1,000-fold. So now if you have a fluorescent molecule whose brightness actually depends on the calcium concentration, you instead of, now you can just image this particular neuron. And now if you look at the gray traces, those are the electric signal. Each little vertical line a single action potential. But then when you look at the brightness of this neuron, you will see this variation of brightness in responding to the change of intracellular calcium concentration. And we call those variations calcium transients, and they can be quantified by the change of the fluorescence value divided by the basal fluorescence value, or delta F over F. This way, using those genetically encoded calcium indicators, you can actually monitor the activity of neurons in vivo in an awake behaving animal. For example, in this movie, each donuts are actually individual neurons. Those are the cell bodies, the soma of the neurons. You can see that those flashes, those are the calcium transients, indicating this particular neurons were active at that particular time. And not only you can look at this activity on a single cell level, you can also see the activity on a single synapse level. So here we express those proteins into a dendritic spine and giving the animal different kind of visual stimulation. You can see that the different spines respond, have calcium transient for different visual stimulation, suggesting that they are selecting this information differently. This sounds wonderful. Two-photon fluorescence microscopy allows us to both look at the processes on a single synapse level and also allow you to look at many neurons that form the neural circuit at the same time. Can we now finally understand the brain? The sad thing is that we cannot actually imaging the whole brain with that kind of resolution. In fact, even for mouse brain, which is a lot smaller, with the cortex, which is a very superficial structure in the brain, only being about 1 millimeter thick, using a conventional microscope, you can only have nice resolution at the very superficial part of the mouse brain because of two very simple physical phenomena, and that is aberration and scattering. First, I'm going to focus on aberration. You all know for a day like today, if you look out a window when it's raining, you will have a rather distorted image of the environment. Although ideally on a nice day, what will happen is that the focus lens will direct all the light onto the same spot, which is the focal spot on the CCD camera, and give you a nice image. But the water film on the window actually distorts the wavefront of the incoming light so that you do not have a nicely focused light rays. And this is called aberration. When you try to imaging mouse brain in vivo, you have the same problem. Ideally, you want to focus your excitation laser for two-photon fluorescence generation into a diffraction-limited focus, which means all the light rays should intersect at the same point, and they should also have the same phase, so they can constructively interfere to give you such a diffraction-limited focus. But because the refractive index or the optical property of the biological sample is actually usually different from the immersion media for which your microscope is designed, what those inhomogeneity does is that it will change the direction and the phase of the light ray, so that at close to the focus, they do not actually intersect at the same point. And in the places where they do intersect, they do not have the right phase relationship, so that they might destructively interfere. As a result, what you have now is instead of a very tight diffraction limit sized focus, you will have a large and very weak focus. And this is the aberrated focus. In other words, even though you have a very fancy microscope that you spend all your time build to make sure it's totally perfect, once you look at biological sample, the sample-induced aberration will now make it impossible for you to get sharp images. And one way to get around this is that if you could pre-distort your light rays, for example, here by changing the tilt of those light rays so that it will cancel out the sample-induced aberration, and near the focus, you can then recover a diffraction-limited focus. Now everything is good again. The problem is, how do you know which kind of distortion you have to put on the incoming wavefront to cancel the sample-induced aberration? So for many years, astronomers has been fighting a similar problem when they try to look at the stars using a ground-based telescope. So ideally, just like the cartoon I showed you before, what you want to happen in your telescope is that you have the planar wavefront coming from the distant star, and the telescope will transform this planar wavefront into a spherical wavefront, and you have a diffraction-limited image of the star on your camera. But before the light can actually hit the telescope, it has to go through atmosphere, where you have airs of different temperature and different humidity, which actually means they have different refractive index. And what it does is that they actually distort the wavefront of the starlight so that you cannot form a diffraction-limited image of the star anymore. What makes it even harder for the astronomers are that this aberration is also temporally dependent because atmosphere fluctuates very rapidly. So when you look at the individual star, you will see these very variable images that are moving around. This is actually the physical basis of the sun, twinkle, twinkle, little star. So whenever you're seeing the sun, you're actually talking about a rather, a deep physical fact on the light propagation. So the solution that astronomers come up with are very elegant and simple. So what you do is that now you have this distorted wavefront of the starlight coming into the telescope. But you can bounce off this light off a mirror whose surface shape you can control. And then you can pick off some of this light and send it into a device called wavefront sensor, which basically sends the wavefront. And then you can send this information back to the controller of this mirror, and you can do it really quick on the millisecond scale, so that after the light bounces off this mirror, you will have a flat wavefront, which will give you a diffraction-limited star, image of the star. So this is the same star, but then with the adaptive optics turned on, you can clearly appreciate the improvement of resolution. And if you compare the brightness of these two things here, they are normalized. But if you compare them on the same scale, you can see that the signal actually, the brightness increased 20 times. So this is the same image from the movie I was showing you before. With adaptive optics, you can really see the stars in the galactic center. So this sounds wonderful. Can we just use this geometry to correct the aberration in the mouse brain? There's a little problem, because if you look at the geometry here, you see that you have to measure the wavefront after it has been distorted. If you look at the geometry in the two-photon fluorescence microscopy, this means that you need to measure the wavefront somewhere close to the focus, which means, and this is based on the relative size of the wavefront sensor and the mouse, you can put the wavefront sensor into the mouse, but it's not in vivo imaging anymore. So you won't see much activity or mental activity from the mouse. So in other words, we have to think of a different way to measure the wavefront from a sample where you cannot directly measure them. And the physical picture we come up with is actually very simple. So I already told you the problem with non-corrected images. So, but the first thing we basically need to do is that we need to do this so-called tilt correction, meaning if you separate the laser into multiple light rays, you tilt each light ray so that they actually intersect at the same point. Once you achieve that, you just need to make sure that all the light rays are in phase. So we call that piston correction. And once you have these two steps done, you basically now have a diffraction-limited focus. It turns out the second step, piston correction, is easy. Because what you are trying to do is just to make sure all the light rays are in phase, and you guys are physicists, you know it's actually very easy to do. We could just steal this method from astronomers. If you think that the distorted wavefront is actually continuous, you can simply calculate what the phase have to be once you know the tilt. But sometimes the aberration is not continuous. For example, in the Keck telescope, the primary mirrors are segmented mirrors, and you actually have to face the mirror to make sure they are all in phase. And then this kind of assumption doesn't work anymore. but we can do a simple interferometric experiment where you can use a reference ray and then step the phase of a separate ray and monitor the signal change. So what you want to find out is the additional phase you have to add to this ray so that they will be constructively interfere. And this is very simple. And if you look at that equation, what you need to find out is basically, if you could keep varying the phase you add to this ray, what you are trying to find out is the phase of a cosine function. And there's many, many ways to do that. One of the nice ways is to use Fourier transform. The reason you want to use Fourier transform is that now you can actually measure the phase of multiple light rays at the same time by modulating them at different frequency. So I'm not going to show you detail, but I just want to say this is a solved problem. Usually within 10 milliseconds, we can already determine what the phase should be. So what we are left with is how do we make sure that all the light rays will intersect at the same point in a biological sample. So the first method that we developed when I was a postdoc in Eric Betzig's group is what we call the single segment illumination. The physical picture is rather simple. Imagine you have these 3 light rays coming in to generate a focus. The left and the right ones are aberrated by the sample, but the center ones are not. And you get an aberrated image from your focal plane. Now imagine that you can turn off 2 light rays and only use 1 light ray to take a 2-photon image. For the light ray that is not aberrated, this light is already reaching the desired focal point. So you will not see any shift from your reference image. But for this light ray, which is missing the desired focal point a little bit, you will see a little bit displacement. And if it displaces in the other direction, you also know, how the relative position of the light ray at the focal point. So once you know how much the displacements are, and you know the focal length of your objective, you can simply calculate how much you need to tilt each ray to make sure they overlap at focus. And experimentally, that's where the name comes from. How do we do this? So imagine that this circle is a real pupil of your microscope objective. So that's the one side of the objective is very close to the mouse. The other side is the back pupil of the objective. And what we do is that you can shrink your laser beam into a small size and then illuminate different part of this pupil, that's different segment of this pupil, and at each position, take an image, say, of a fluorescent bead or a cell body. And then you can just look at how this image deviated away from the center. From there, you can directly get the aberration of your sample and generate the corresponding corrective wavefront. So this method works very well. It works in vivo. So in this case, we introduce the red fluorescence beads, and we use that to measure the aberration. But now I'm showing you the volume imaging stack of neurons in a mouse brain in vivo. On the left-hand side are the lateral and the top view and the side view of the structure without adaptive optics correction. On the right-hand side, it's after the correction. And here, I actually artificially increase the brightness of the image 2 times so that you can see those structures. So you can clearly see that adaptive optics improve the brightness of your image. But more importantly, it's a lot more easier now to follow the process, the follow individual neuronal processes after correction, which is very hard to do before correction. So this method works, but it has a limitation. Well, most of the method has limitations. And I'm going to tell you the limitation of this method is that when we only illuminate a small segment of the pupil, what you are basically doing is that you are not using the full resolving power of the objective. In other words, the focus you form when you only illuminate part of the pupil become a lot larger compared to when you illuminate the full pupil, which is how you usually use your microscope. If you have a very sparsely labeled sample, that is okay, because you can still very easily determine the shift of the images when you're doing the single segment illumination. But if you have a densely labeled sample, now you have a problem. Here you have some structure that is originally out of focus. You're not really seeing it in your reference image. But then after you reduce the size of the light rays, now it's actually very hard for you to determine how much the image was shifted. In other words, that method works very well, but it does not work when the label you want to use are very dense inside the sample. But there is actually a solution. And this is what I have been working on after I started my own group. in that there's always a very sparse point inside your sample, and that is your laser focus. So we can have a totally different way to measure the sample-induced aberration, which we call full pupil illumination. As I said, an ideal focus is when the light rays hit the same point and have the right phase. So if you can change the tip and tilt, you move the one light rays around the reference focus formed by the other light rays. If there's no aberration, you will get the brightest signal when you do not add any additional tip and tilt to this light ray because it's already going to the focus to begin with. But if there is aberration, then you have to add some additional tip and tilt of this light ray so that it will hit the reference focus and give you the nice interference. So, for example, in reality, we can separate the pupil into 36 region, and then we can get such an intensity variation data for each of these 36 corresponding light rays. But you can see that there's a problem. The signal to background ratio is rather low in this approach because you're trying to detect the focal intensity variation when you move 1 out of 36 of this overall energy in and out of focus. This is particularly acute when you look at the side, the light rays at the side where the variation could be as small as 2%. So now if you want to look at a small variation, that means you need lots of photon. And also because we have to do this measurement one light rays at a time, the overall just make it very small to measure the aberration. It could take 20 minutes to measure what is the aberration in your sample, which is just unacceptably long for in vivo imaging experiment. So how can we make the method better? The first thing we can do is to get rid of this DC background. This is like the oldest trick in physics book is that you could modulate the intensity or the phase or even the polarization of this light ray. So any property that would affect how this light will interfere with the reference focus. And then instead of plotting the DC value of your signal as we were doing before, you can do a Fourier transform, and then you can just plot the magnitude. And you mostly now get rid of this DC background, you will have an image with a lot better noise level. And the biggest advantage of this approach really is that now you could actually measure the tilt required for multiple light rays at the same time by modulating their property at different frequency. So this is a multiplexing kind of method which really increases the speed of this correction. So now instead of 20 minutes, we could modulate 18 segments at the same time, and with a better device now, we could do this aberration measurement within 30 seconds. So, and in reality, it also works very well in vivo. So what you're seeing are a very densely enabled mouse brain, and it's about 200 micron from the surface of the brain. And here we just park our laser focus at this cross, which is the cell body. And you're seeing the lateral and the top view and the side view of the same structure here. And after 2 minutes of measuring the aberration, we get a corrected wavefront, and we can take the image of the structure again. Clearly, you can see a lot of improvement in both signal and resolution. We could look at, for example, the line intensity profile along the different part of this image. And you can see that in this red line, before correction, you mostly see a hazy background, and you don't really see many neuronal processes. But after correction, you can clearly resolve the dendrite of many different neurons. And there's some interesting physics here, is that if you look at this side view along this orange line, you see that there's a cell body here. And you could actually see there is a cell body even before you do adaptive optical correction. But then what you really see that after the correction, you can also detect a few dendrites, which are really invisible before you do the correction. However, these two structures really are experiencing the same type of aberration, but the effect on their imaging quality is very different. And the reason is because when you have an aberrated focus, you have a large, weak focus. Now, if you have a big structure like a cell body, even though at each position your fluorescent signal is a lot lower because the electric field is smaller, but in this case, your focal volume is bigger, so it can actually excite more fluorescent molecules, which partly compensate for the loss of focal intensity. But if you have a small structure, like a dendrite, then its signal really only depends on the focal intensity, where the difference is really, really big before and after aberration correction. In other words, if what you are interested in is in cell body, adaptive optics will help, but it may not be essential. But if what you are interested in are the single synapses where the neurons are making connection and communicate with each other, adaptive optics can actually be essential for the study of their properties. So now we can try to image deeper. We go to about 400 micron into the mouse brain. And you can, before correction, you can see some vague faint lines. Those are the dendrites. And on top of those dendrites, the little protrusions are the dendritic spines I told you earlier about. And we can park our laser focus on this dendrite and measure the aberration. And after we apply the correction, you can see that now we have a much sharper image of those dendritic spines. What is interesting is that if you just look at the line intensity profile along this orange line, you see that before the correction, you don't really see those dendritic spines, but after the correction, they are a lot more easier to appreciate. That is not a surprise, but what is surprising is that once you move away from those dendritic spines, those local structure actually does not have any fluorescence molecule in them. However, you still have this background signal, which adaptive optics is really not making a difference. And this background signal is what's called out of focus excitation. Because the mouse brain is scattering, when you try to image deeper, you have to increase the laser power to compensate for the scattering loss of the ballistic component that form your focus. Eventually, you increase your laser power so much that you do not actually need the confinement of the focus to have efficient two-photon fluorescence excitation. In other words, you will have, if you have any labeled structure at the surface of the brain, you will have this out-of-focus signal. And when this out-of-focus signal overwhelms the in-focus signal, you reach the theoretical limit of two-photon fluorescence imaging. But because adaptive optics only modifies the phase of the in-focus signal and doesn't do much to the out-of-focus signal, we actually do not see any improvement in any increase of this background after aberration correction. Which means now we can actually reduce the excitation power and still have a very fairly nice image, even though it's weaker compared to that particular image, but you still have very sharp features with the advantage that we can actually, in this case, completely suppress the out-of-focus excitation, which means that with the help of adaptive optics, you could actually now image deeper in mouse brain. Speaking of imaging deeper, now we go to about 500 micron inside the mouse brain. What you're looking at Z-stack series in the brain. You can see some vaguely cell body, profile of cell body, and those faint lines are the dendrites of those neurons. After adaptive optical correction, you see that the images are not brighter, and you can also detect a lot more of those protrusions, which are the dendritic spines that are on this neuron. It's probably more easily seen if we zoom into this small area, and I just want to maybe pause it a little bit so you can see. In this case, you have two dendritic spines here, which is really impossible to see before correction. And when we move a few micron down, the same thing, these two spines is just not really invisible before adaptive optical correction. And this is just the individual frame in that movie I was just showing you for you to see how much more we can see, especially in terms of this synapses with the help of adaptive optics. One very nice thing, which makes our life easier compared to astronomer, is that you do not need to constantly correct the aberration in a mouse brain because the mouse cells are not turbulent, unlike the atmosphere. And also, when you do the correction at a small area, the same correction actually can improve the image quality over a fairly large area, which means that when we're trying to do biological experiment, my postdocs and students don't have to spend all their time keeping measuring the aberration. Instead, they measure it once at the beginning of the experiment and then can spend hours to do the real biological imaging part of the experiment. So I've spent all that time telling you how we can measure the aberration without directly sensing the wavefront, because we cannot really put the wavefront sensor inside the sample. But how about try to measure the wavefront outside the sample? Now imagine if you have a local, very small guide star, a fluorescent structure inside the sample, And the fluorescence will go out of this sample and then will be aberrated by it. And in principle, you can measure the aberration of this wavefront directly, right? Because nothing is in the way. Would this work? Can we measure the wavefront aberration outside the brain? To answer this question, we need to kind of know how an ordinary wavefront sensor works. So in this particular image, we have a guide star, light goes out, everything is perfect. Then you have this parallel light, have a flat wavefront coming out of your microscope. And if you have a CCD camera, each light ray will form a dot image of itself. And since wavefront is 2D, what you get is a regular 2-dimensional array of dots. If you have aberration, what will happen is that the direction of those light rays will be changed, and they will hit the CCD camera at a different location. When you look at the 2D dimension, you will see this shift of those local points away from the desired aberration-free region. And from that, you can basically directly calculate the aberration that your sample is experiencing. But there is a problem. When you look down on the surface of a mouse brain, you see that you do not see the chin of the mouse because the brain is actually strongly scattering. So for you to get the wavefront information from the guide star is actually very difficult. For example, if we have a yellow fluorescent guide star that is 500 micron below the surface of the brain, on the camera, instead of having very sharp dots, you have a very fuzzy image because scattering basically scrambled the wavefront information. But those images was taken with visible light. When we look at the brain, we're using our visible vision. If you use a longer wavelength light, so if you go from a yellow light into a red light, only 40 nanometer shift, now you can already see rather nicely the individual dots on your wavefront sensor. And you can go all the way to near infrared, where the scattering goes down, quite a bit. And you can actually have a very nice quality wavefront sensing pattern on your camera. So we did this experiment where we introduced a near-infrared dye into the mouse brain in vivo, and this is the imaging stack we can get after correction. And here we're at 600 nanometer. So you can clearly see the dendritic spines on those neurons, and compared to if you do not use adaptive optics, so those are the images of those stretches of dendrites without AO correction. You can see that now those synapses really is not possible to see beforehand. We also did a different experiment when we were working on that project, is that instead of using two-photon fluorescence microscopy, we use confocal. And usually confocal is a single photon process, and people usually say it doesn't work well in mouse brain because it's rather scattering. But what we can see is that with adaptive optics, we can have a very decent confocal image of the brain in vivo. Here, the label, we are labeling the membrane of a neuron. And you can see that after adaptive optical correction, you can almost see the hollowness of these dendritic spines because the resolution of confocal is quite a bit better than two-photon. And in another example, you can see the exclusion, so those are the dendrites, and you can see those local dark region, which we think are the mitochondria subcellular structure inside the dendrite. But the reason I'm showing you this image is that when you look at the surface of the brain, all the way down to 100 micron, where you can already have lots of cells there, scattering is actually not that bad, which means that it actually have lots of promise to try to do super resolution imaging in the brain in vivo, and that's something we'll start to working on right now. And for this kind of imaging, aberration is even more essential. So we need to use adaptive optics to help us to get super resolution information from the mouse brain in vivo. So many times when I talk to biologists, they always ask me, so how deep can you image? I just want to say that this is a question that's impossible to answer because it depends on what you want to image. Do you want to image cell bodies that are filled with lots of fluorescent dye, or very small 1 micron size dendrite? Are you trying to look at a green fluorescent signal or a red one? Because the red fluorescence is less scattered, so you can image deeper. Also, whether you want to look at the sample tomorrow, because you could put in lots of energy into the brain, and if you don't need to see it tomorrow, it's okay. But if you put too many energy in there, then the cell will actually die. It will become unhealthy. But what we can say is that with the most popular genetically encoded calcium indicator, we can now functionally image the input and output of the mouse primary visual cortex with synaptic resolution. And what is also very important for any kind of technology development is how easy it is to use your technique, especially for biologists. And I would like to say that, and I will show you an example towards the end of my talk, how we could actually use it to answer biological questions. But before then, I just want to quickly tell you how people deal with the scattering problem, which arguably is much harder to deal with than aberration. So with scattering, no amount of adaptive optical correction can help you to image a structure that is at the very bottom of the mouse brain. For example, this structure called the lateral hypothalamus is about 5 millimeters down. Because even if you can effectively excite fluorescence here, the emitted fluorescence will be scattered all over the place on its way out of the brain. You're just not going to get enough signal. So one of the solutions that people come up with are the so-called endomicroscopy, originally by Mark Smither's group and Walt Webb's group. So here, what you have to do is to have a compromise. If you can tolerate a little bit of invasiveness in your sample, then you can embed a very micro optical probe inside the brain, which can then realize the focus of a conventional objective inside the biological sample very deep. And typically, such a micro-optical device are made of this green lens, it's a gradient refractive index lens. So unlike the conventional lens, which has a uniform refractive index, this lens has a higher refractive index in the center. And you could have just a single snap of this lens, but it will have the focal function just like a conventional lens. But there are two problems with the endomicroscopy approach. The first one, especially for the high resolution endomicroscopy, is that the imaging field of view are very small. So this is just some nice optical engineering. For example, this is a design where you can see the individual synapses. It is only about 1 millimeter in diameter, which seems to be very, very small, and is actually about 1,000 times smaller than a conventional objective of similar resolving power, similar numeric aperture. But if you look at the effective field of view, you see that little blinking dots there. This is how big you can actually image inside a biological sample. And the reason is because this objective lens is so small, you cannot fit in all the other lenses to correct the off-axis aberration, which is very easy for us to do with adaptive optics. So we can do that and increase the field of view tremendously. For example, this is a cell body away from the center of the imaging field of view. After correction, you can now resolve those very fine neuronal processes. And this is a much larger field of view before and after adaptive optical correction. And you can compare those regions on the corner, and you can clearly see now we can image a much bigger area, which is essential. Because another problem with this approach is the damage. So because even though this is about 1 millimeter in diameter, the whole mouse brain in this dimension could be just about 5 millimeters. So putting such a big structure inside the brain is still too much. You're removing too much brain circuit for the mouse to be function properly. So usually when people want to look at very deep structure, they do not use such a high resolving power green lens. And usually they can reduce it a little bit to make the probe much smaller. But what people usually do is that you don't want to directly embed this probe in the brain because they are rather expensive, and this surgery has a pretty high failure rate. So instead, they put in a guide tube and then only put the green lens in when you're imaging the brain. And the conventional guide tube is very big. So the volume of the guide tube can be about 3 times the volume of the green lens. So in other words, you're removing all those structure unnecessarily. And we designed a very fine, very thin walled guide tube, which only have the increase volume by 50%. And this is how this guide tube looks like, and this is how it looks like when we embed it in the mouse brain. And what we also investigated is that when the inflammation reaction induced by this implantation of the guide tube will go down. So this is just some antibody labeling to look at the scars inside the brain. You can see that 2 weeks after implantation, there's still lots of scars around the implants. But 4 weeks after the implementation, you actually now have a pretty reasonable sample to look at. And this is how the whole engineered system looks like. And with this system, now you can look at, this is an imaging stack, the neurons in this very deep nuclei, which controls feeding, drinking, and anxiety. And you can also look at the function by measuring the calcium transients, as I described to you before. For example, for this particular structure, we actually see that many neurons have quite a synchronized activity, which is different from a different structure that we looked at called substantia nigra, where the neuron shows very different activity pattern. So with that said, I just want to tell you one example of how we can actually find new biology using the adaptive optical method. So because I was trained as a physicist, for me to understand the circuit, we need to understand what are the inputs of the circuit, what are the outputs of the circuit, and how each component inside the circuit processes the information it receives. And the circuit that I'm interested in is the so-called primary visual cortex. So when you look at this monitor right now, the visual information first goes into your eye, gets to the retina of your eye, and from there goes to a structure called thalamus. In this particular case, a nuclei called LGN. And from there, the information is sent to the striatum cortex or the primary visual cortex. And when we try to look at the activity of those neurons, what we do is that we present a visual stimulation to one eye of this mouse, usually in the form of a grating that is drifting along different direction. And then we open the brain on the other side and look in to see how the neuron respond to this visual stimulation. So the cortex structure I told you earlier before actually are formed by many different layers. And it is known for many years that the neuron in the primary visual cortex are selectively firing towards visual stimulation of different orientation. So let me just walk you through about how this means. If you look at this neurons 3 and 4, what you see is that for neuron 3, it will only give you this calcium transient, meaning their firing action potential, when the grating is horizontal grating. And neuron 4 will only respond when the grating is vertical grating. In other words, the cells in your brain actually are selectively activated by the edges and the orientation of the edges. And as you can see, in any kind of visual scene, orientation tells you a lot about what is happening. And furthermore, some neurons also show this direction selectivity. In here, that the neuron only fire when the grating is moving in one direction, but not when it's moving in the opposite direction. And we all know how sensitive our visual system is for motion. If you go into a party to meet your friends, you go into the room, there's 200 people there. If you have to look at their face one by one to figure out where your friends are, it's going to take you some time. But if your friend sees you and he waves at you, and you can very easily pick up where he is because our visual system is really primed for detecting motion. So for many, many years, people have think that this kind of orientation and motion sensitivity is strictly the property of neurons in the primary visual cortex. So in the classic model, the thalamus that LGN nuclei I was telling you about send the information into layer 4 of the primary visual cortex, from where the information then goes to layer 2, 3, and then we'll go back down to layer 5, and from there to the other part of the brain. And in the classic model, and it's a classical belief in the field, I would say, is that the inputs into the visual cortex are not orientation selective, meaning the neurons sending the information to V1 do not actually select for orientation. And the orientation selectivity is strictly a property of the computation that the cells are doing inside the visual cortex. However, even from the very beginning of vision research, people has noticed in all the model systems they've looked at that some of the neurons in the thalamus actually are already orientation and or direction tuned. The question is, do they actually send those information to the visual cortex? And if they do, how would that affect the feature selectivity in this region? And we can use imaging with the help of adaptive optics to answer this question. So first, we need to image the input into the primary visual cortex, which requires to image in the axon terminal of those thalamic neurons. So we can express the calcium indicator in the thalamic neuron and then look at their projection, their axons inside the primary visual cortex. With the help of, and so basically, well, just to remind you, we're looking now at the bouton, the presynaptic part. With the help of adaptive optics, we can image those bouton, those 1 micron, 2 micron structure very deep inside the mouse brain in vivo without any appreciable degradation of imaging quality because we correct all the aberrations. And when we look at their activities, we're looking at the calcium transient in response to visual stimulation, we got a big surprise. There are certainly boutons that equally active for different orientation, but there are also multiple boutons show this orientation selectivity, and some of the boutons also show the direction selectivity. And when we look at many mouse, we found that more than 50% of the boutons, meaning more than 50% of the input into the visual cortex already selective for orientation. And this actually really depends on the fact that we use the adaptive optics. So this is a calcium transient signal we get without and with adaptive optical correction. So you can see that without correction, our data is a lot weaker because of the brightness concern. As you can see here, if we have a 2 micron fluorescence beads that are under different thickness of cranial window, or a thin cranial window, but without adaptive optic correction, and after adaptive optic correction, you can see that the brightness of this 2 micron beads is about 4 times weaker compared to that. So that explains why we see this brightness difference. In other words, if we look at all the bouton we see, without correcting for aberration in this thick window scenario, we will say only 30% of them are responding to visual stimuli. But if you correct the aberration, now 90% of them will be responding to visual stimuli. But even more importantly for this particular project are the tuning curves. If we do not correct for aberration, you will think only 7% of the bouton are orientation tuned, which is basically saying it's not that orientation tuned. But if you correct it, you will see about, again, 50% of them are tuned. The reason is because of the size change of the focal volume. So if you have a perfect diffraction limited focus and you want to know the tuning property of this white structure, you can see that you can pretty well isolate its contribution in the signal. But now if you have an elongated focus, your signal will be contaminated by all the other boutons which may have different tuning preference. Therefore, you will have a much broader tuning curve. So now we know what the input this circuit is receiving. We can ask how each component, how the orientation information are represented at each component of this circuit. We could use the same technique. We can characterize the neuron in layer 4 that are receiving the input, the neuron in layer 2, 3, and the neuron in layer 5. And now we also routinely measure layer 6. And just similar to before, we have the direction selective population, and we call the population that are non-direction selective axis selective. And from their tuning curve, we can decide what are their desired movement axis or direction. And we can do that for both the input into the circuit. So this is a thalamic bouton input at different thickness inside the mouse visual cortex. It's very surprising in that if you look at the distribution, most of the direction selective input actually prefer a particular direction. And when you look at the cortical neurons, you see that this bias in the input can still be detected in the selectivity of the layer 4 neurons. And what is even more interesting is that this particular direction bias are detected in the output of the circuit, that is the layer 5 neuron. And this actually tells us that maybe there are actually direct connections between the input and output of the circuit, which is a little bit strange, right? Obviously, you want the cortex to process your information. Why do you want to have the direct input? So this is actually not a very far-stretched proposition. Such a direct connection has been seen using electrophysiological method, using electrical recording method in many other different systems. And we also have now some direct evidence of where the connections are made, but I'm not gonna bore you with all the biology. I just want to summarize what we found in this particular project. With the help of adaptive optics, we can now interrogate the input and the output of a neural circuit at synaptic resolution. What we found is that the input into visual cortex, contrary to the general belief for the past 50 years, are already orientation tuned. And there's also a direct connection between the input and output neuron of this circuit. I just want to make a little bit comment. This particular direction is a posterior to anterior motion. And if you're a mouse, where you spend lots of your time in your life actually running away from predators, any kind of movement that's coming out from behind are important information. This is probably why even at the input in the circuit, you already have this information very heavily represented. Now, since we have a direct synaptic connection between the input and output of the neuron, which means that this particularly important information may directly get out of the visual circuit without having to be processed and get distributed to other brain areas, like watch out, something is coming. I don't care whether it's a football or it's a fox, but you better pay attention and ready to run. So right now we are trying to keep going into this pathway to see whether that's exactly what's happened. So I'm gonna just summarize. So most of the work are done by several members of my lab. The adaptive optical work was mostly, also the under microscopy work mostly done by Chen Wang with help from Renil. And the neurobiology work, the axon imaging story, was spearheaded by Wen Zhishun with the help of Zhongqiao Tan. So currently my lab are half physicist and half biologist. And I found this is really, really useful for the technology development aspect, because I do not develop a technique that my biologists wouldn't want to use. And we also have the initial work I reported was done in Eric Betzig's lab, and we also have some the direct wavefront sensing work was also done in collaboration with his postdoc Kai Wang. And we have lots of help, lots of collaborators that help us with questions and sample preparation. And I work in this place called Genilayer, which is a very small group and focused direction. And all my colleagues give me a huge amount of help in helping me to appreciate the difficulty and the intriguingness of how brain work and help us to direct our future research direction. So I have to thank them a lot for their support. And thank you for your attention. So questions?
The photos are not that yucky. I suppose because there's no blood on it. Okay, any question?
So you're looking at a part of the
Yes. So there's one project actually I didn't describe, is where we study the UV vision of mouse. So people actually, even neurobiologists who study mouse, they don't actually know that the mouse visual system are very sensitive to UV light. So we went in and tried to understand it with the goal to try to look for color opponent cells, because there's some reports saying that the mouse actually have color vision. Given that they are such a wonderful model system for neurobiology, we try to see whether it could be a good model. What is a little bit sad is we only find 1% of the neuron have that property. It's there. We also see some developmental differences. So in human baby, the sensitivity to blue, which is the short cone vision, develop later compared to the longer wavelength cone vision. So in mouse, we actually see the same thing, that the cortical neurons, their response to UV stimuli is a bit delayed. And we published it actually, but we didn't look at we did look at the input a little bit too, look at how the thalamic buttons encode color information. But what we basically saw is that mouse visual system encode UV information in very similar fashion as they do visible information. So we do not actually expect much difference, and we didn't see any difference either.
So when you're using the guidance source, the directed wavefront, so is there no appreciable distortion, like wavelength dependent?
That's a very good question. You would think there would be a wavelength dependence, right? So aberration basically is caused by optical path difference, and if there's any kind of dispersion, and also your wavelength is different, so there should be a difference there. That's actually the advantage of the direct wavefront sensing using near-infrared dye, because when we use a dye that is emitting the fluorescence at 840 nanometer, and our excitation light is at 900 nanometer, any effect of dispersion is going to be very small. So we didn't really see anything significant. But that's a very good question.
You use the recombinant layer 4 and
Right.
find this strong bias towards activating orientation selective because it's really striking. And I wonder if you could use the green adaptive technology to look down the LGN and really confirm the snow body levels.
So there was some extracellular recording data in the LGN where some of the paper is very similar to ours. 50% of neurons are orientation or direction tuned, but they didn't have enough neurons to really do the kind of distribution that we see. So you could use the green lens to do that. People also have tried to just remove the cortex on top at Canaway's group, cortex and hippocampus, and they could image the top 50 micron of the neurons. But from our perspective, the phenomena is extremely robust. We can only look at one imaging session. Actually, we can get the same trend because we often have 300 botons there. So in a way, we overdid our measurement by 2 orders of magnitude. So it's a very robust thing. So we're basically not going to really try to confirm it because for us, it actually doesn't matter how many cell body have certain selectivity. Since we care about the input, where counting synapses are more useful, we think, than counting the cell from which the synapses come from, because different cell may have different number of synapses in one.
Not necessarily true for the glucon to match the soma because you have feedback on the
Right. So that's something that we really wondered about, and of course the reviewers ask about them. What we know is that we tried awake animal, we tried anesthetized animal. For when we anesthetize the animal, the neurons are not as responsive, but for the responsive population, the tunings are still the same. So that's not a direct answer to your question, but it is suggestive to us that the tuning are not very state dependent.
This is all cone driven, it's all bright.
Right, so we were using very bright stimulation. We use the stimulation that everyone else in the field are using, which are more cone driven.
I was just wondering, I don't know if it's practical to do it, but I was wondering if you see the same kind of selectivity if you that under darker conditions.
Right. So we did this luminance dependence experiment for the UV project. What we see is we can go down really weak, and at least the neurons in cortex still have a pretty similar activity level. I actually think that in the field, people basically use too bright a stimulation. It is kind of abnormal. There's behavior observation that the mouse, when they do visually driven behavior task, they actually perform better, they learn faster if you reduce the stimulation. But that's something that the field hasn't really explored, and we haven't either. But it's conceivable that there will be some difference there. Yeah. So are you asking whether the same method can be used for other biological samples? So absolutely. Even for cells, if you use very high resolving power objective, 10 micron away from your cover glass, you start to see aberration. For all the model biological system, cyanogans, zebrafish, there's all sample-induced aberration. And this method, and all the other different type of AO method can basically is very useful if you want to imaging anything that is above the cover glass by 10 micron. Absolutely.
How much variation do you see going from mouse to mouse? So when you set up your microscope for one mouse, does it work pretty well?
For aberration?
Yeah. For aberration.
It always works well. Yeah, usually the nice thing about this method is it's very straightforward. sometimes
Can you use the same correction?
right. So no, that's a short answer. And the longer answer is that it's not a simple refractive index mismatch, even though I made it sound very simple, because the curvature, the brain can have different curvature, and even in the same mouse, at a different region or different depths, you can see this variation of the aberration. But what you could do is that for the same mouse, if every time you can mount the mouse exactly the same way, in principle, you only need to do this once. So my collaborator, Carol Spoda lab, they actually built this very fancy, tip-tilt motorized stage where they dial in the position of each mouse. And we are trying to make our AO correction device very easy to add on to their microscope. And that's something we're going to try to see that maybe you only need to do it once. And every time when you grab the same mouse, you don't need to do the correction anymore. That should be stable, I believe.
If there's no more questions, thank you, speaker again.
Thank you.